This is the rotavirus. Now, the rotavirus is a leading cause of diarrhea and dehydration, most commonly affecting children under 5 years old. Its occurrence can vary depending on a country's mortality rate. Studies have shown that in countries with higher mortality rates, rotavirus infections tend to occur around 38 weeks of age, while in countries with lower mortality rates, it is more common around 65 weeks of age. Rotavirus is mainly spread through the fecal-oral route. So when a child comes in contact with feces containing the virus, and no hand washing was done afterwards, then their hands can easily become contaminated. If then the child puts their contaminated hand into their mouth, that's when the virus enters the body and starts its journey. Rotavirus is a non-envelope virus, and non-envelope viruses are able to withstand harsh environments. It also has multiple protein layers. As you can see here, one of these include viral protein 4 or VP4, which forms these orange spikes. So, yup, all of these orange spikes are called VP4. On the other hand, this yellow layer is called viral protein 7 or VP7. And together, they create this thing called a capsid layer. This structure allows the virus to resist the low pH levels of the gastric juice in our stomach, enabling them to survive. It will then make its way past our duodenum to target the jejunum and ileum. There, it specifically targets mature enterocytes because they are abundant in the jejunum and ileum. It can recognize and then bind to receptors that are located on the surface of the enterocyte. Using its VP4 spikes, the virus attached firmly to these receptors, only for it to then penetrate the cell. This causes the cell to perform endocytosis, to be able to engulf the virus. The rotavirus then sheds its outer VP4 and VP7 layer upon entering the cell. The virus is left with the layer below it, which is the viral protein 6 or VP6 layer. The virus then produces mRNA that is released into the cytoplasm to make viral proteins. One of these viral proteins is non-structural proteins such as NSP4. This is especially troubling because NSP4 is an enterotoxin. This enterotoxin then damages the enterocyte itself, leading to the impairment of the villi. If we were to zoom in to the apical surface of the enterocytes, then this is what we'll find. The enterocyte actually plays a crucial role in the bridging of absorption. On the left side is the lumen, while on the right side is the blood vessel. Enterocytes has a lot of transporters. A few of them include antiporters such as the chloride bicarbonate exchanger, which normally absorbs chloride into the enterocytes while secreting bicarbonate into the lumen, and the sodium hydrogen exchanger, which normally absorbs sodium into the enterocytes while secreting hydrogen into the lumen. But because the enterocyte is damaged, then both of these exchangers experience dysfunction, which means these antiporters stop working properly. Not only that, this also affects the SFTR channel that usually helps with the secretion of chloride, but instead, it causes an excessive amount of chloride to be secreted. At the same time, the brush border is damaged and cannot produce enzymes such as maltase, lactase, and sucrase. Without these enzymes, these saccharides cannot be broken down into monosaccharides. So for example, if a child were to drink formula milk, then this can become a problem because formula milk is rich in lactose, and lactose is a disaccharide. So the thing is, when lactose reaches the small intestine, it is supposed to be broken down by one of the brush-burner enzyme, which is lactase. Lactase helps break down lactose into glucose and galactose, which are monosaccharides, and monosaccharides help with absorption. What should be considered is that there is one working exchanger left, which is a symporter called the SGLT1. This transporter helps absorb glucose and sodium all together. But the thing is, this symporter can only work if both glucose and sodium are present. If one of them isn't present, then this exchanger will shut down. And normally, when this absorption happens, water will follow. But in this case, neither water nor lactose can be absorbed because, again, lactose is a disaccharide and disaccharides cannot be absorbed. It can only be absorbed when it gets broken down into monosaccharides. And water can only be absorbed when the SGLT1 is working properly. And remember, the SGLT1 is dependent on the presence of glucose, which is a monosaccharide, and the presence of sodium. Due to the fact that neither ions nor lactose or other disaccharides cannot be absorbed, this explains how malabsorption happens. And malabsorption, with an increased secretion of the chloride, causes the consistency of the stool to turn watery. And this is usually found in diarrhea. Undigested lactose then passes into the colon where it encounters Escherichia coli, or what is commonly known as E. coli. E. coli is a lactose fermenting bacteria, and this causes the lactose to undergo a fermentation process. So not only did the stool become watery, it also becomes acidic due to the fermentation process. When this acidic stool comes into contact with the skin inside the diaper, it can cause irritation, especially when the diaper isn't immediately changed. And this irritation explains why children that experience diarrhea from the rotavirus tends to have red rashes around the perianal and anal area. And that is all. So, that is how the pathophysiology of the rotavirus leads to diarrhea, malabsorption, and associated symptoms in patients such as red rashes and acidic stool. Well, that is all for today. Thank you so much for watching, and I hope this explanation was helpful. See you next time. Goodbye!